All right, everyone, welcome in. Today, we're diving deep into the world of interferon receptors.
Yeah, how they help our bodies fight off viral infections.
You want to understand how your cells combat those pesky viruses, right?
Right.
Well, it all starts with these incredible receptors.
It really is remarkable how these receptors act like a cellular defense system,
Yeah.
detecting viruses and triggering a cascade of responses to neutralize them.
Okay, so we're not just talking about one type of receptor, right? We've got type 2 and type 3 interferon receptors.
You got it.
What makes them different?
And each type has its own unique way of dealing with viral invaders. They activate different signaling pathways within the cell, ultimately leading to the expression of antiviral genes.
Oh,
It's like having a specialized team of defenders, each with its own set of weapons.
so these antiviral genes are like the blueprints for building the proteins that actually fight off the virus.
Hmm. Exactly. Think of it like this. The interferon receptor is the alarm system, and the antiviral genes are the instructions for building the weapons to combat the intruder.
That's a great analogy. Now, I know we have some scientific excerpts lined up that delve into the nitty-gritty of how this all works.
Right?
I'm curious to see what we uncover.
Let's dive in. One thing that stood out to me was the discussion of glycine in one of the excerpts. It's fascinating how something as simple as this tiny amino acid plays a crucial role in the spinal cord.
Glycine. Isn't that just a building block for proteins? What's it doing in the spinal cord?
Well, it turns out that glycine is the primary inhibitory neurotransmitter in the spinal cord. You see, for every signal telling your muscles to contract, there's a corresponding inhibitory signal from glycine ensuring smooth, controlled movements.
So it's like a braking system preventing our muscles from going into overdrive.
Precisely. Imagine trying to pick up a delicate object.
Yeah.
Without glycine, your movements would be jerky and uncoordinated.
So glycine is basically the unsung hero of coordinated movement, keeping us from spilling our coffee everywhere.
Exactly. What I found particularly interesting was the concept of a closed, desensitized state of the glycine receptor.
A closed, desensitized state. What does that even mean?
Well, if the receptor is exposed to glycine for a prolonged period, it can become less responsive,
Okay.
almost like it's taking a break.
So it's like a safety mechanism to prevent the cell from being overwhelmed by a constant signal.
That's one way to think about it. It highlights the incredible complexity and self-regulation within our cells. And speaking of complexity, let's shift gears and talk about cardiomyocytes, those amazing heart muscle cells.
Okay, what about them? I'm all ears.
They illustrate perfectly how cells can be incredibly specific about how they respond to signals, even when the same messenger molecule is involved.
Wait, so cells can differentiate between different types of messages even if the messenger is the same?
Yeah.
How is that possible?
Let's take cyclic AMP, or CARAMP for short. It's a common signaling molecule involved in various cellular processes. Cardiomyocytes have different receptors that both trigger the production of CARAMP, beta-1 adrenergic receptors, and EP4 receptors.
So same messenger, different receptors, got it.
Yep.
But what's the catch?
The catch is that stimulating beta-1 receptors makes the heart contract harder. But stimulating EP4 receptors has absolutely no effect on contractility.
So the same messenger molecule can trigger different responses depending on which receptor it binds to. That's fascinating.
It challenges the classical view of global cellular signaling, where CARAMP would flood the entire cell and have the same effect regardless of the receptor. This suggests that signals might be localized to specific areas within the cell, like tiny compartments or microdomains.
Microdomains.
Yeah.
So it's like the cell has its own internal zip codes, ensuring messages get delivered to the right department.
That's a great way to put it. And this is where A-kinase anchoring proteins, or AKKPs, come into play.
AKPs, those sound like they're about to drop the hottest new track. But seriously, what do they do?
Think of AKPs as the organizers of the cellular orchestra, ensuring that each instrument plays its part at the right time and place. They bind to protein kinase A, or pKa, which is an enzyme activated by KMP.
So instead of KMP causing a global flood, these AKPs are making sure pKa is strategically positioned to carry out specific instructions.
Exactly. And there are over 50 types of AKPs in humans, each with its own unique targeting domain that determines its location within the cell.
Wow, that's incredible. It's like having designated stage managers for each section of the orchestra, ensuring that the music comes together harmoniously.
And if we zoom in even further to the molecular level, we find this fascinating structure in AKPs called an amphipathic helix.
Okay.
It's like molecular Velcro binding pKa to specific locations.
Molecular Velcro. Okay, you've got to explain that one.
Well, this helix has a water-repelling side and a water-attracting side. The water-repelling side binds to a specific motif on pKa, keeping them firmly attached.
So it's like a perfect molecular fit, keeping pKa in the right place to carry out its instructions. Amazing.
And what's even more interesting is that AKPs preferentially bind to type 2 pKa, suggesting that this specific type of pKa might be crucial for the localized signaling we see in those cardiomyocytes.
So type 2 pKa has a backstage pass to the AKAP concert, while other types are stuck in the general admission area.
You could say that. It really highlights how even the tiniest details can have a big impact on how cells function.
Absolutely. But before we get too lost in the molecular details, let's switch gears again and talk about thyroid hormone receptors. They sound like another key player in this intricate cellular communication network.
They are indeed. And they illustrate another fascinating aspect of cellular signaling, the ability of a single receptor to act as both a gene silencer and a gene activator.
Wait, so they can turn genes on and off? How does that work?
Yeah. It all comes down to the presence or absence of thyroid hormone. In its absence, these receptors bind to DNA and actually repress gene expression, keeping things quiet. But when thyroid hormone is present, it binds to the receptor, flipping the switch and activating gene expression.
So it's like a light switch, but with thyroid hormone acting as the hand that flips it on or off.
That's a good analogy. And this delicate balance is crucial. Mutations that disrupt this mechanism can lead to developmental defects and diseases like resistance to thyroid hormone, or RTH.
So in RTH, the switch is either stuck in the off position or can't be flipped to on effectively.
You got it. This can have widespread consequences for the body, impacting everything from growth and development to metabolism.
It's incredible how these tiny molecular switches can have such a profound impact on our overall health. But, we've been talking a lot about different types of receptors and signaling pathways. It makes me wonder how those interferon receptors we started with fit into this whole picture.
That's a great question, and it brings us back to the main focus of our deep dive today, those incredible antiviral defenses. Remember, interferon receptors are like cellular sentinels constantly scanning for signs of viral invaders.
Right, those cellular gatekeepers we talked about earlier. They detect the virus and then sound the alarm, triggering a cascade of events to fight back. But how does that actually happen? What's going on inside the cell once the alarm is raised?
Well, it all starts with interferon molecules, which are like tiny messengers released by infected cells. Think of it as a distress signal. These interferons bind to interferon receptors on neighboring cells, alerting them to the viral threat.
So it's like a cellular neighborhood watch program warning everyone to lock their doors and stay vigilant.
Exactly. This early warning system gives those neighboring cells time to ramp up their defenses before the virus can reach them.
Okay, so they've received the warning signal. What happens next? How do they actually build up their defenses?
This is where things get really interesting. Interferon receptors activate a complex chain of events known as the JAK-STAT pathway. This pathway is like a molecular relay race, passing the signal from the receptor to the nucleus of the cell where the DNA is stored.
A molecular relay race. I like that. Who are the runners in this race?
The first runners are enzymes called Janus kinases, or JAKs. They're attached to the interferon receptor and get activated when the interferon molecule binds.
So the interferon acts like the starting pistol, setting the JAKs in motion.
Precisely. And the JAKs then pass the baton in the form of phosphate groups to another set of proteins called STATs, or signal transducers and activators of transcription.
So the JAKs phosphorylate the STATs, giving them the energy to run the next leg of the race.
Exactly. And once those STATs receive their phosphate boost, they pair up and head straight for the nucleus where the DNA is stored.
They're on a mission to deliver a vital message. But what do they do once they reach the nucleus?
They bind to specific regions of the DNA and activate the expression of those antiviral genes we've been talking about.
So they're like tiny construction crews turning on the machinery to build the cell's defenses.
That's a great analogy. And those defenses come in various forms. Some antiviral proteins directly interfere with viral replication, like disrupting the virus's ability to copy itself. Others boost the immune system's response, helping to identify and destroy infected cells.
So it's a multi-pronged attack hitting the virus from different angles.
Exactly. And what's remarkable is that the interferon system can adapt to different types of viruses. It's like having a universal defense system effective against a wide range of threats.
No wonder it's so crucial for our immune system.
Yeah.
But you mentioned earlier that there are different types of interferon receptors. Do they all activate the same JAK-STAT pathway?
That's a great question, and it highlights another layer of complexity in this system. While they all utilize the JAK-STAT pathway, Different interferon receptor types can activate different combinations of JAKs and STATs.
So it's not just a simple on-off switch. There's nuance and variation depending on which type of receptor is involved.
Precisely. And this fine-tuning allows for a more tailored response depending on the specific type of viral threat the body is facing. For example, type I interferon receptors, which are found on most cell types, are often the first responders to a viral infection. They activate a specific set of JAKs and STATs that trigger a broad antiviral response, helping to contain the infection early on.
So they're like the general alarm alerting all the cells in the area to prepare for a potential attack.
Exactly. Now, type 2 interferon receptors are a bit different. They're found mainly on immune cells and play a more specialized role in coordinating the immune response. They activate a different set of JAKs and STATs that enhance the activity of certain immune cells like natural killer cells and macrophages.
So they're like the special forces called in to target and eliminate infected cells.
Sure. That's a great way to put it. And finally, we have the type 3 interferon receptors, which are relatively new players in this field. They're found primarily on epithelial cells, which line the surfaces of our body like the skin and the lining of our respiratory tract. They also activate the JAK-STAT pathway, but trigger a slightly different set of antiviral responses compared to type I interferons.
So they're like the border patrol guarding the entry points to our body.
Precisely. It's fascinating how each type of interferon receptor has its own unique role to play in this intricate defense system.
This is all incredibly complex, but so fascinating. It really makes you appreciate the incredible power and precision of our immune system. But you mentioned earlier that interferon receptors also play a role in cancer immunotherapy. How does that work?
That's a great question, and it highlights the potential of harnessing the power of interferon signaling to fight cancer. You see, cancer cells often try to evade the immune system by suppressing interferon signaling.
So they're trying to silence the alarm system, preventing the immune system from recognizing and attacking them.
Exactly. But researchers have found that by boosting interferon signaling, we can enhance the immune system's ability to detect and destroy cancer cells.
So it's like turning the alarm volume way up, making it impossible for those cancer cells to hide.
You got it. And this is the basis for many cancer immunotherapy approaches that use interferon-based therapies.
Wow, that's incredible. It sounds like interferon receptors are truly a key player in both fighting infections and potentially revolutionizing cancer treatment.
Absolutely. And as researchers continue to unravel the intricacies of interferon signaling, we can expect to see even more targeted and effective therapies emerge in the future.
This has been such a fascinating deep dive, but before we wrap things up, I'm curious about one more thing. While we were going through those sources, I came across something called negative thyroid hormone response elements, or NTREs. What are those all about?
Yes, NTREs. They add another layer of complexity to the already fascinating story of thyroid hormone receptors. Remember how we said these receptors can act as both gene silencers and gene activators, depending on whether a thyroid hormone is present or not?
Right. The presence of thyroid hormone usually flips the switch from off to on activating gene expression.
Well, with NTREs, it's the opposite. When a thyroid hormone receptor binds to an NTRE, the presence of thyroid hormone actually leads to gene repression.
Right, so it's like a reverse switch. Instead of turning the lights on thyroid hormone, actually dims them.
Precisely. It highlights the fact that gene regulation is rarely a simple on-off switch. There are many nuances and variations depending on the specific genes and regulatory elements involved.
So it's less like a light switch and more like a dimmer with a complex control panel.
That's a great way to visualize it. And it reminds us that even though we've uncovered a lot about these systems, there's still so much more to learn.
Absolutely, and that's the beauty of science. There's always another layer to peel back, another mystery to unravel.
Exactly. It keeps us curious and constantly learning.
Well said. So as we wrap up our deep dive into interferon receptors and antiviral defenses, what are the key takeaways you want our listeners to walk away with?
I think the biggest takeaway is the sheer complexity and elegance of our immune system. These tiny molecular players, interferon receptors, JAKs, STATs, antiviral proteins, they all work together in this incredibly orchestrated dance to protect us from viral invaders.
It's like a microscopic symphony happening inside our bodies, constantly defending us from threats we don't even realize are there.
And what's amazing is that we're only just beginning to understand the full scope of how this system works. There's still so much more to discover, and with each new discovery comes the potential for developing new and more effective therapies for a wide range of diseases.
Absolutely. From antiviral drugs to cancer immunotherapy, the potential of harnessing the power of interferon signaling is truly remarkable.
And it all starts with understanding the fundamentals. how these receptors work, how they activate different signaling pathways, and how those pathways lead to the production of those crucial antiviral defenses.
So for our listener who's been on this deep dive with us today, what's one thing they can do to learn more about this fascinating field?
I encourage them to keep asking questions, read articles, watch videos, talk to scientists. The more we learn about the intricate workings of our immune system, the better equipped we'll be to protect ourselves and those around us.
Well said. And on that note, we've reached the end of our deep dives into interferon receptors and antiviral defenses. We hope you've enjoyed the journey and that you've come away with a newfound appreciation for the incredible power and complexity of your own immune system.
Until next time, stay curious, keep learning, and remember, knowledge is power when it comes to safeguarding our health.